Hello, my dear students. I hope all of you are doing very well. So, my name is Abhijit Devnath, and from today, I'm going to start a lecture series on novel drug delivery systems. So, those of you who are actually reading in B Pharm 7th semester, this lecture series is going to help you a lot because in this entire series, I have tried to summarize entire concept of novel drug delivery system in a very simplest manner. If you're going to do some research project in FND or drug delivery system, again, this subject or this my video is going to help you a lot. So this is the first lecture, introductory lecture, and it is very simple. I have tried to make this lecture much more simple, and all the basic necessary concepts I have tried to cover in the first lecture. So at first I'm going to tell you what is drug and formulation. Then I'm going to tell you the drug discovery and development process. And after that, I'm going to tell you about the challenges in drug delivery. My dear students, there are lots of challenges in drug delivery, and that's why this subject NDDS actually evolved. So the challenges we can solve by using NDDS. So to tell you how we can solve all the challenges that are happening in drug delivery by using NDDS, at first we're going to learn the basic things. So let me start with the drug and formulation. So the simplest question, what is drug? The most simplest definition of drug will be a drug is a chemical substance used to treat, cure, prevent, or diagnose a disease or to promote well-being. My dear students, all of you have suffered during the time of COVID. Somebody might got infected, and some of us might be safe during that time. So the ultimate thing which actually saved us, it is drug, the drug like remdesivir. So in simplest definition, a drug is a chemical substance which actually used to treat, cure, prevent, uh, or diagnose a disease or to promote well-being. But sometimes in the subject called pharmacology, we actually tell a different definition. In pharmacology, a drug is a chemical substance which administered to a living organism produce a biological effect. So if you administer a drug molecule into a bacteria or a virus, and if it is harmful for bacteria and virus, it is going to kill it. So it will be a called, if this phenomenon will called as a biological effect. So that's why in a much more appropriate way in pharmacology, we are telling this definition. But sometimes if a drug is producing some neurological effect, so you have to tell in a different context. So a drug is a substance that causes a change in an organism's physiology or psychology when consumed. So these are the most popular three definitions I have actually sorted and I have tried to tell you. So according to the need, you can use any of this definition. Now from where we're going to get this drug molecule? First, we can get those drug molecule from the nature, like aspirin, penicillin, or morphine. I hope all of you are aware aspirin is the first such drug molecule which is actually we have derived from nature and we have synthesized that drug molecule in our lab. So you can also get the drug molecule from synthetic sources. You can synthesize, you can make a new drug molecule in your lab. But if you have already a natural product, if it is having a good biological activity, you can make some necessary changes in its chemical structure. So you can call it as a natural derivatives. So you take a drug molecule from nature, make some necessary changes in the drug molecule structure in your lab, in a synthetic lab, and if you give the drug molecule to the patients, it will be called as a natural derivatives. Now, what should be a quality of a good drug? First and foremost, a drug should be safe. It should be safe. It should not produce any kinds of unsafe or undesirable effect. It should possess the high efficacy and it should not show any kinds of adverse reaction. And yes, it should not possess any kinds of side effects. And it should show effective ADME. What is ADME? I hope all of you have studied absorption, distribution, metabolism, and excretion. And it should be non-toxic. Apart from this, a drug should be therapeutic. Obviously, it should be therapeutically effective, and it should bind with the specific target. So all of you are aware that in our body there are more than 20,000 different types of proteins, and the concept right now what we are actually following, it is called one drug, one target. One drug, one target. It means the drug should go and bind with its particular target, specific target in maximum time called protein. Maximum time we're actually targeting the proteins in your body. And I have told you there are more than 20,000 different proteins. So the drug should be bind with its specific target. It should not bind with the other targets. So here you can see this is a protein and this is the active site of this protein, and the drug is very nicely binding with this protein, and it should not possess any kinds of off-target binding. If it is possess any kinds of off-target binding, we'll be observing various kinds of side effects, adverse reaction, and it should be stable at the active site. And yes, this drug molecule should be free from any kinds of side effects, like whatever the common side effects we're actually observing in our the mostly used drug molecules like vomiting, nausea, sedation, insomnia, aggression, diarrhea, dizziness, anxiety, and confusion. Such things should not be happen without drug molecule. And yes, the most important thing is this particular slide. So this is the base of our this subject called NDTS, because in next whatever the new technologies we're going to learn in our NDTS, every time I am going to refer particular this slide. So your drug should be within this therapeutic window. The concentration of drug should be within this therapeutic window. So when you consume a drug molecule at 0th time, after sometimes the drug molecule concentration is going to increase. It should reach within the therapeutic window, and it should remain there for at least a good amount of time so that it can give desired therapeutic response. If the duration of action is very high, then you have to take a low, that is a low number of doses, less number of doses. And yes, it should have, this concentration should be within the therapeutic window. If the drug concentration should unable to reach the minimum effective concentration, it will not show any kinds of therapeutic action. It is called as a subtherapeutic. And yes, if you consume huge amounts of drug, huge amounts of your drug molecule, so what will happen? It will show some side effect or adverse response. Why? Because it is crossing this minimum maximum effective concentration. So when we design a drug molecule, we have to be careful that its duration of action should be high, and it should be within, its concentration should be within the therapeutic window. Now, all of we have, I have talked multiple times about this term called formulation. So what is formulation? So for example, I have a drug, like this is the structure of a drug. In sometimes we call it is as an API. API means active pharmaceutical ingredient. Okay, drug API, it's the same thing. So sometimes we call it as an active pharmaceutical ingredient, because ultimately this active pharmaceutical ingredient, it is going to the site of action. Yeah, it is going to the site of action, binding with this protein and making some bonds like hydrogen bonds or van der Waals or pi sticking with this particular protein and showing its desired therapeutic action. So that's why sometimes we call it as an API, active pharmaceutical ingredient. So my drug molecule, and when we bind it or when we add it with some excipients, we actually getting formulation. Now, what are the common example excipients like binder, coating agents? If you make some tablet doses form, in this context I'm actually telling, like binder, coating agents, preservatives, sweeteners, colors and flavors, solubilizers, disintegrants, fillers, diluents, glidants or antiadherents. These are the things we can call in the terms of excipients. So ultimately these things will be forming our formulation. Now, so sometimes we actually confuse between dosage form and formulation, है So I also try to tell you, I'm going to tell you that what are the relationship between the dosage form and formulation. So dosage form are the pharmaceutical drug products in the form of in which they are marketed for use with a specific mixture of active ingredient and inactive component. So active ingredient means this API, and inactive means the excipients. Okay, so in a particular configuration and appropriated in the into a particular dose. So all of you have studied in our B.Pharm first semester that based on the route of administration, we can classify into oral, sublingual, parenteral, conjunctival, and many of the other doses form. These are the most common example of our oral doses form like tablet, capsule, solution, syrup, elixir, etc., etc., and sublingual like our tablet, troches, lozenge, etc., and parenteral like solution, suspension, conjunctival like contact lens, inserts, and ointments. These are the most common doses from based on our route of administration. Now, let me also connect this thing with the drug delivery system. So to understand the delivery system, it's a method or process of administering a pharmaceutical compound to achieve a therapeutic effect. I can also tell in a different context that a delivery system is used to often refer where doses from releases the drug and deliver it to a targeted organ. So, for example, this is a tablet. Yeah, this is a tablet, and if you zoom this tablet into 10,000 million x, ultimately you'll be getting our this drug molecule. Okay, so what I've shown you in the previous slide, that I have drug molecule, I have mixed my drug molecule with some excipients, then I have formed tablet. And what is the goal, what is the goal behind this? Because it is like a delivery. So whatever the excipients actually mixed, these are actually working like a delivery. This is a capillary, or this is a blood vessels, you can say. So ultimately, this delivery system are actually going to bring our drug molecule at the site of action. Here you can see this, these are various cancer cells. These are various cancer cells, and this is a cancer causing protein. So my drug is actually going here and binding with this cancer causing protein and curing cancer. So ultimately you see here you are observing only this drug molecule, this API. Here you can see no any excipients like the binding agents, lubricating agents are actually present. So the desired therapeutic action actually producing by only our the API. So that's why the excipients are actually playing a role which are actually a delivery mechanism. So the excipients, whatever we have mixed, this is actually helping to deliver the drug molecule to our site of action. So I can tell the relationship between the dosage form and delivery system. So the dosage form is the final substance, the marketed or the physical appearance of our final thing what are actually getting in the market. And within this, our delivery system actually produce. So this is the relationship between the dosage form and delivery system. Maximum time we actually use this word interchangeably. So dosage form is often used to refer to a physical appearance of a drug like tablet, capsule, syrup or other things, whereas the delivery system is often used to refer to the way dosage form releases the drug and deliver to the targeted organ. I hope it is very much clear to you after watching this picture and this picture. So ultimately this is my drug molecule which is actually entrapped within this tablet, and which tablet having the excipients, and the entire tablet actually helping our this drug molecule to reach at the site of action. Now, after learning the drug formulation and what is the relationship between the drug delivery and the dosage form, now let me tell you the drug discovery and development process. So at first, I will tell you what are the steps involved in drug discovery and development. So today, by using computer-aided drug design, we discover drug molecules. There are a few other ways also possible, but in 21st century, we actually use computer. Right now, we are using machine learning, AI, and deep learning based techniques to discover drug molecules. And mostly the docking simulation, DFT, DMA toxicity, and all the things are actually doing right now in computer. So ultimately, you discover a drug molecule through modern approaches, and after that, you're going to do some preclinical test. You're going to test the safety, efficacy, and the effectiveness of the molecule on some rat, rabbit, or any kinds of species. If your drug molecule is showing better, a good therapeutic effectivity, then you're going to make some formulation. We call it as an early formulation development. Our NDDS concept is actually coming here. If your drug molecule having good therapeutic effectivity, but it has low solubility or it has low permeability, at that time we can use NDDS based techniques. And if the drug molecule is fine, it does not having any low solubility, low permeability or any kinds of other issues in delivery mechanism. So at that time, we can simply make some tablet, capsule, syrup, and we can deliver the drug molecule. We have no need to take some novel formulation or novel drug delivery system. So if we get the proper after making this early formulation development, what is the next step? The next step is clinical studies, clinical trial. There are three phases of clinical trial. So after successful in your clinical trial, if what do you do? In US, you're going to file a new drug application to FDA. In India, it is called CDSCO. You're going to tell them that yes, please see my data, my drug molecule with this formulation or with this dosage form, it is showing proper therapeutic effectivity in this number of clinical trial records, and it does not having any kinds of toxicity, side effects or adverse reaction, and it is curing a particular disease. Now please approve me to bring the drug molecule into the market. So FDA or in India, it is called CDSU, they will analyze your safety data, and after that they will give you the approval. So after approval, what is the next step? The next step is industrial scale production. So in industry, you're going to produce that molecule in a huge quantity, and after that, you have to recruit few people as a marketing representative who are going to visit the physician's chamber, and they're going to inform the physician about your new drug molecule and its proper therapeutic effectivity. And they will start prescribing, and your drug will be going to come to the market. And after that, when patient actually start consuming, if a drug having some safety issue, then pharmacovigilance will come to the picture. So this is the entire steps or entire landscape from concept to market. In nutshell, this part, the first step is called drug discovery. This part from preclinical studies to industrial scales production, this entire thing actually coming under drug development, and the new drug application, some parts of industrial scale production, the marketing and pharmacovigilance are actually coming under drug regulatory affair. So you should be careful when we use different words. Drug discovery and drug development both are different. Okay. Now, this is a very nice picture or nice data where you can see that how many drugs are actually get approved by US FDA with since 1993 to 2022. So BLA, what is BLA? BLA means biological entity like antibodies. So the dark green things actually whatever I have highlighted here, these are actually biological entity like antibodies. And NME means new molecular entity. Okay, like our small molecule, this green thing, this light green thing actually small molecule. You can see that every year huge number of actually drugs are actually approved by US FDA. You can see in 1996 highest number of drugs actually approved by US FDA, it is 47. Then there is a sharp decline, and there is a sudden rise in the US FDA's drug approval. So what is the reason behind this sudden rise since 2005 to 2021? The reason is the advancement of technology like computer aided drug design, molecular docking, molecular dynamic simulation, DFT, drug likeness, ADME, toxicity, and also machine learning, artificial intelligence, and deep learning based techniques. So all the things are actually helping to increase to get the better drug molecule, which molecules are actually showing much more better therapeutic effectivity in clinical trial, and FDA is actually approving the drug molecules. So now the biggest question you're going to might be thinking that how many approved drug molecules are available. So it is very difficult to tell the exact number because there are different regulatory agencies are available in different countries. In US it is called FDA. In India it is called CDSCO. In Europe, Russia, China, Japan, they have their own regulatory agency. So it is very difficult to tell the exact number that how many drugs actually are approved by FDA. So let me take the reference of drug bank. So as per drug bank data, total number of approved small molecule based drugs are actually 2,748. But if you see the Campbell data, it is 2,992. I am recording this lecture on today's 22nd August 2023. So this is the number of today. This is the number as of now. Now, if you want to know that how many drugs are actually approved by FDA, what are the latest drugs are actually approved by US FDA. So let me take you to this link, this website of US FDA. So I will share this link in your in the description of this video. So if you click on this link, you will come to particular this page. So at bottom, if you come to the bottom, you'll be observing that what are the new drug molecules actually approved by FDA. So I am recording this lecture on 23rd of August 2023. Just recently, 5 days back, US FDA approved one, I think, monoclonal antibody for particular disease. You can read in your free time. Here all the diseases details are actually available, and what are the drugs actually approved by US FDA are available here. This is the brand name, and this is the active ingredient in the generic name. Okay, so you may visit. So if you come to this page every year, so these are the list of molecules which are actually approved in this year. This year, as of now, they have approved 35 drug molecules. And if you want to know what are the drug molecules approved in last year, you can again click here and you can see that what are the drug molecules. And if you want to see year wise, like 2021, 2020, you can explore by yourself. Now, so now, after learning these two segments, the drug and formulation, we also have seen that what are the process steps involved in drug discovery and drug development. Now, let me take to you what are the challenges in drug delivery. That is the most of the common issues are associated with the failure of drug. So, my dear students, at first, I do not want to discourage you, or what I can say. This slide might be slightly what I can say discouraging, because you can see that it is it is near about 1.8 billion dollar, and more than 10 years actually required to bring a drug molecule into market. Okay, so it's a very complex and difficult task. Many of you might be thinking that, sir, during COVID time, we have seen that the COVID-19 was actually appeared at the end of 2019, or at the beginning of 2020. And within just 6 and 7 months, we got new drug molecule called remdesivir and few of the new other drug molecules. Yes, this is true. For COVID-19, we discovered the drug molecule and brought the drug molecule to the market in a very short period of time. But there is something called accelerated approval, and due to the pandemic scenario, US FDA actually approved this drug molecule based on their safety data. Okay, but if you see in the context of other drug molecule, so to bring a drug molecule from concept to market, it takes more than 10 years, and you have to spend near about 1.8 billion dollar. Okay, so we can understand that how complex and how challenging the task is, from discovery to develop and to bring into the market. And if I tell you more, so the success rate, this is 1/10. It means out of 10 clinical trial, or out of 10 particular project, only one project is going to successful. And here you can see the list of clinical trial flops. These are the molecules which actually flopped during the clinical trial in last year 2022. You can see the indication like breast cancer, Alzheimer's disease, and geographic atrophy, and many of the other diseases. They have tried their best. These are the name of the company like Sanofi, the Roche and Genentech, Roche again Pfizer. You can see the big pharma companies name here. They have tried their best to cure those diseases by their new drug molecules, but they failed in clinical trial. So why they failed in clinical trial, and how we can solve this? So this is why we're actually learning this entire lecture. So let me also tell you there is something called valley of death. Many of the drug molecules actually failed particular in this reason. So first we identify some target, then we go for hit to lead optimization, then preclinical development, where we do the study on animal, and then we go for the phase 1, phase 2, phase 3 clinical trial. So maximum of the drug molecule actually failed in particular this reason. That is phase 1 and phase 2 clinical trial or preclinical trial. So that's why it is called as a valley of death. Now, what are the main challenges? Why actually drug molecules actually get failed? One of the reason is solubility, then poor permeability, then poor bioavailability, poor drug absorption, inadequate distribution, deposition of drug into adipose tissues, and stability of the drug. These are the major reasons why our drug molecules actually failed, and this is the major challenge in our drug delivery. But yes, we can solve those challenges, because right now we have better technology which is coming under our novel drug delivery system. Now, my dear students, you have to also understand that if the drug molecule is showing some toxic effect, then we cannot solve this drug molecule by only NDDS. Now, I also have to tell, we can categorize the entire drug molecule into four different segments, like class 1, class 2, class 3, class 4. What is class 1? High solubility of the drug and high permeability of the drug. These are the best kind of molecules which you can take orally. And yes, there are chances the drug molecule might be having low solubility or high permeability, or high solubility, low permeability. And it might be having low solubility and low permeability. So this is called as a BCS classification of drugs. So if we see the market, so the class 4 molecules having 10% of drug molecules are actually BCS class 4 drug molecules, and 35% of the marketed formulation or candidates up to have to 10% actually BCS class 1 formulation. You can take those drug molecule orally, but for BCS class 4 molecules you cannot take them orally because they have low solubility. If you take the drug molecule through your oral route, when it will go to your stomach, it is not going to dissolve. There will be no any disintegration, there will be no any dissolution, and it is not going to absorb through your intestine. Okay, and due to its low permeability, it will also again not permeate to your various cell membrane, tissues, etc., etc. So ultimately, this is going to create a big challenge in our field. Here is a big, very nice representation where you can see the increasing chances of non-oral dosage from being required. So from BCS class 1 to class 4, BCS class 1 drugs are actually simplest solid dosage form. So you can take them orally. But as soon as you go to lower, the non-oral dosage form actually increase. Non-oral dosage form means like injection, your other kinds of parenteral dosage form, which actually we don't like to take through injection or other routes. Now, here I have tried to summarize much more with some examples. So, if a small molecule, if it is coming under the BCS class 4 or any of the other segments like class 2 and 3, so the challenge is actually controlling the PK parameter. PK means pharmacokinetic. In particular, like half-life, biodistribution, or exposure and maximum concentration. There are other challenges like improving solubility, permeability, whatever I have just told you right now, target development, and yes, reducing off-target toxicity. I have listed few examples. You can pause the video, you can read it, and you can remember for your examination purposes. So I'm just trying to encourage you so that you understand why we are actually learning particular this subject called NDDS. These are the few challenges, and we can solve these challenges by using NDDS. So let me summarize. So the poor solubility, poor permeability, poor bioavailability, poor drug absorption, inadequate distribution, deposition of a drug in adipose tissues, and stability of the drug. These are the creating the biggest challenges in our drug delivery. So being a student, you might be asking, is there any solution? I will simply say yes. Now, what is the solution? The answer is novel drug delivery system. That's why you have spent with me since last, I don't know when I've started this lecture, I think 10 to 15 minutes, or might be some more minutes you have spent with me to understand that why we're actually learning this subject called NDTS. These are the challenges, or these are the problems are actually associated with our this thing called drug discovery and drug development scenario. Now, if you ask me that what is the definition of novel drug delivery system? For today or for this lecture, I will simply tell you that a novel drug delivery system is a method or process that introduces therapeutic substances in the body more efficiently and effectively, enhancing their efficacy and reducing the side effects, so that the drug molecule may show its proper therapeutic effectivity, and whatever we have shown in our previous slide, that it should be a show as a good drug, good quality drug. There is no any toxicity, there will be no any adverse reaction or free from side effects, and it will be go at the active site of the target protein and strongly bind, and it will be cure particular this disease. So that's why the NDDS actually come to the picture. So please stay with me. In our next lectures, I'm going to tell you what is NDDS, what are the various segments of NDDS, and how we are actually using NDDS for making better drug delivery. So that's it from my side for today. So thank you, my dear students, for being with me. So keep learning, keep shining. I'm also available in various social platforms. I have a website called theabi.in. You may visit my website. I will again also upload this PPT in my website. You may visit, you may scroll, you can change the slides, and you can see and enter the slide in my website. I'm also available on Facebook, Twitter. Right now, Twitter become X. That's why I have added the X icon here. And I'm also available in Instagram. You may search on the social media platform with this username, the Abhijit DN. You'll be getting me. So we shall catch there, and best of luck to all of you. And I hope we're going to start. We're going to learn a lot of things, and I wish that all of you going to shine in your life. And this subject NDTS going to help you for making your research project and help you much to shine in our this pharma field. So thank you all of you. Thank you for being with me. So see you in the next lecture. God bless.